In biosynthetic studies in plants, the tracer technique is a powerful tool used to track the movement of specific molecules or elements within the plant's metabolic pathways. By introducing labeled or marked compounds known as tracers into the plant, researchers can follow the incorporation and transformation of these tracers as they are assimilated and metabolized by the plant. The tracer technique allows scientists to investigate various aspects of plant biosynthesis, including the synthesis of complex molecules such as carbohydrates, proteins, lipids, and secondary metabolites. It provides valuable information about the flow of carbon and other nutrients through different biochemical pathways, shedding light on the plant's metabolic processes and how it utilizes resources for growth, development, and defense. Common tracers used in plant biosynthetic studies. Stable isotopes. Stable isotopes of elements like carbon, e.g., (13C), nitrogen, e.g., (15N), and sulfur, e.g., (34S), can be used as tracers. These isotopes are non-radioactive and can be easily detected and quantified using advanced analytical techniques like mass spectrometry. Radioactive isotopes. Although less commonly used due to safety concerns, radioactive isotopes can also serve as tracers in plant studies. For example, (32P) can be used to trace the movement of phosphorus within the plant. Isotope-labeled precursors. Isotope-labeled precursors are compounds containing stable isotopes that are incorporated into specific biochemical pathways. For example, (13C) labeled glucose can be used to study the synthesis of carbohydrates. Heavy water. Deuterium oxide, D2O. Heavy water can be used to trace water movement within the plant and study various metabolic processes dependent on water availability. Applications of tracer technique in plant biosynthetic studies. Photosynthesis. Tracer techniques are used to study the assimilation of carbon dioxide during photosynthesis and the flow of carbon within the Calvin cycle. Isotope-labeled carbon dioxide, (13CO2), can be introduced to understand the rate of carbon fixation and the distribution of carbon among different metabolites. Amino acid biosynthesis. Tracers can be used to investigate the biosynthesis of amino acids. For instance, labeled nitrogen, (15N), can be employed to track nitrogen uptake and utilization in amino acid metabolism. Secondary metabolites. Tracer techniques can be applied to study the synthesis of secondary metabolites, such as flavonoids, alkaloids, and terpenoids. This provides insights into the regulation of these compounds and their ecological roles. Lipid metabolism. Tracers can be used to examine lipid biosynthesis, including the production of fatty acids and storage lipids like oils. Plant pathogen interactions. Tracers can help understand how plants respond to pathogen attack, enabling researchers to track the synthesis and distribution of defense-related metabolites. Challenges and considerations. Tracer experiments require careful design and optimization to ensure accurate interpretation of results. The choice of tracers should be specific to the metabolic pathway under study. Consideration of plant species, growth conditions, and experimental setup is essential to obtain reliable data.